Well, welcome everybody to our oak pest and disease workshop. We have some exciting speakers for you here today. Starting off, we're going to have Dr. Bea Noboa-Berman, who I think most of you already know, University of California Cooperative Extension Advisor for both Orange and Los Angeles counties, specializing in urban forestry. Her research and extension program is focused on finding appropriate management strategies for urban forest pests that affect Southern California trees, like the invasive shot-hole borer beetles and the gold-spotted oak borer. Following her, we'll have Dr. Shannon Lynch. She is an assistant professor at UC Davis's Department of Plant Pathology. And she also is the immediate past chair of the California Invasive Species Advisory Committee. And it was a recent recipient of the Western chapter of ISA's award for arboriculture research. It's worked extensively in California, studying invasive shot-hole borers and various other pests and plant diseases. Dr. Curtis Ewing is an entomologist and senior environmental scientist with CAL FIRE's Forest Entomology and Pathology Group. Covers the Cascade and Northern Sierra region. He's been doing a lot of the work on the Mediterranean oak borer, and he's also co-chair of the California Firewood Task Force. And then lastly, Dr. Chris Sjogren is an environmental horticultural advisor for UC Cooperative Extension in LA and Orange County. And as an advisor, he provides expertise in pest management of ornamental plants and water use issues.
All right, so I'm going to talk about gold-spotted oak borer. I see some faces that I recognize from last Wednesday. Sorry, guys, this is even a shorter version of that, so you won't see anything new. But hopefully, the rest of you can learn something from this. So, gold-spotted oak borer is from the family of the jewel beetles, Agrilus auriculatus. It's this little beetle about 3/8 of an inch. So think about it like the size of wild rice, as long as a wild rice grain. And it has 3 different hosts: coast live oak, California black oak, and canyon live oak. Coast live oak and black oak are the preferred hosts, but it can also attack canyon live oak. It's just not a preferred host. So it will attack it if it's there, but it will, if it has an option to attack any of these two, it's going to go for those. How does GSOP attack a tree? Basically, it lays their eggs in the crevices of the bark. There it comes like a teeny tiny larvae, and that teeny tiny larva will start eating or chewing on the bark and making its way through the bark to reach the cambium. And in the cambium, it's going to start feeding on both the cambium, the xylem, and the phloem. As it feeds, it makes all these galleries that kind of grows. It chews and poops and they just keep filling them with poop. And then what happens is some fungus start colonizing that frass, which is how scientists say insect poop. Start colonizing that frass and it turns black. And so if you could see, so this dark staining here in the cambium will be, if you cut in here and you look at the cambium layer, this is how it will look. And it looks kind of like not much of a pattern, like a plate of spaghetti. And that's the pattern for GSOP, is that there's no pattern in the galleries. Other bark beetles have a specific pattern, like S-shaped or beautiful particular shapes. In GSOP, the pattern is that it's just a squiggly thing, mess, and it's everywhere. So just as a simplified life cycle, we'll start here with the adult emerging. And as I say, they will lay their eggs. So they emerge kind of in the spring. And then they'll, after a few days, they will feed on the canopy. And then after a few days, lay their eggs. Those eggs hatch. We have several instars of larvae feeding on the cambium and phloem and xylem and compromising all that vascular system of the tree. And eventually, that's what actually is affecting the tree. Then they stay in prepupae stage during the winter. They overwinter as prepupae, and this shape as kind of looks like a hairpin, right? So this hairpin configuration, we like to call it, is how they stay until it starts getting warm and they pupate, and then again, they emerge, leaving this, very characteristic D-shaped exit holes, and we start again. So this whole cycle takes 1 year. The current distribution and potential distribution of G-sub, this is where it is right now. So this is in Southern California, will be this area here. And all these points in red are areas where we found G-sub. It started here in San Diego, and that's where we found the most. But then we kept finding small focuses all over Orange County, Riverside, San Bernardino, LA. And we know, in some cases, we know for sure that the way they arrive to these new locations is through firewood, infested firewood. In some other cases, we don't know for sure, but that's what we suspect. And the problem with this pest, so this pest is from Southeast Arizona. And so it is native to the United States, but it's not native to California. So the oaks here are not, did not co-evolve with the pest. And so they don't have the defenses against the pest. There's not, the natural enemies are not here. So there's not things to keep them in check, right? The climate here and in Arizona is slightly different. So it's the dry season here is in the summer, and the dry season in Arizona, where they're from, is in the winter. Yeah, is in the winter. The other way around, the wet season here is in the summer, and the wet season in Arizona is in the wet season here is in the winter. Excuse me. Wet season in Arizona is in the summer. So when the beetles are doing the most damage, the larvae feeding during the summer, the trees in Arizona are getting water. The trees in California, when the beetles are doing the most damage, they're just being drought stressed. So it adds up. So it's another contributing factor for trees here in Arizona. And oh my gosh, in California, I'm so sorry. I hope it didn't confuse you. I'm gonna stop talking about those two states. And this other map with the colors from green to red shows, considering where the hosts are available, the potential range for G-sub to spread, right? And so we can see that there's many areas that can be very affected. Now, this is a picture of San Diego in the Scansum-Watai. This is ground zero where we found it. It's been there for, well, right now, a couple of decades. But as you can see, there's several dead trees already in this area. About 40 to 45% of the canopy was affected by G-sub. And that's what happens when you have this pest and you're not able to do any kind of management. And the kind of symptoms we can see in Coast-Side oak, when a tree is just starting to get infested, you might see no crown differences, right? This crown, even though the picture is a little bit dark, but that looks like a normal crown, but it's already infested with G-sub. So you could have trees that don't show crown symptoms, but they're still attacked. And as the infestation progresses, we start seeing some crown thinning, like in this case. And then when they get more and more infested, we start seeing dieback of entire branches. That's in the case of Coast-Side oak. In the case of black oak, so Coast-Side oak is evergreen tree, right? But black oak is deciduous. It loses the leaves in the winter. So when it's infested with G-sub, the first thing that we see is that the leaves will turn brown, but it will hang on to the leaves. It will not drop the leaves. So that's first kind of like red flag. And the next thing that will happen is that when the next round of leaves comes out, they come out a lot smaller. So it has this kind of weird pattern. That's another symptom in the crown that you can look in black oaks. The other thing that happens a lot is this staining and cracks on the bark. That's pretty common in black oaks as well, especially when they get kind of heavily infested. So to go over other evidence of attack, we talk about dieback and crown thinning. We talk about staining in the bark. The beetles have been chewing on the xylem and phloem and kind of like stops the movement of water. So water kind of accumulates in a place in the trunk and then starts seeping out. And it looks dark because it gets stained with all the tannins from the tree. You can also see some evidence of woodpecker activity, especially in heavily infested trees. The woodpeckers will go and eat the pupa that are kind of on the very outer part of the bark. And if you actually kind of take an axe and looking under the bark, you can see the galleries. And this is, well, except for this one, which you won't go and chop into a tree unless you know that that tree is a goner, right? But the woodpecker, the staining, and the dieback are things that we're going to, maybe they're gonna let us know that there's something going on with that tree. But the real telling that this tree is infested with GSOP and not something else is the D-shaped exit holes. This is your key. You need to find the D-shaped exit holes. If you see dieback, that could be basically any of the things we're gonna be talking today. It could be a fungal infection, it could be a different kind of pest. Same with the bleeding, the cracking. But if you see D-shaped exit holes in an oak in California, for now at least, we know it's GSOP. Now, will we see if a GSOP is infested if we can only look at the first, 6 feet of the trunk? And the answer is yes, especially if it's heavily infested. So if you look at this graph in here, the y-axis is how many holes, or the count of the hole density, and the x-axis is the height in the bowl, right? And this is in meters. So to give you an idea, 2 meters is about 6 feet. And then you can see the trees that are lightly infested will have, low amount of exit holes kind of all over the trunk, right? From 0 to 6 meters, it's kind of low counts everywhere, but you will see. So between 0 and 2 meters, you will see some exit holes. But especially in the heavily infested, which are the circles, the black and the white circles, these are trees that are heavily infested, and you can see that the majority of the holes are concentrated in the first 2 meters. So the answer is yes, you can look at the base of the tree, right? As high as you can do it. It depends on who's looking. It's like, the first 6 feet or so, or 8 feet, and you can be pretty sure that if it's a heavily infested tree, you're for sure going to notice. And if it's a lightly infested tree, you most likely will notice something. Now, as I said, GSOP spreads mostly in firewood, and that's because they're not really great flyers. And so actually when they will disperse, they would just go to the nearest tree or like maybe a few trees around. But we see that it's dispersing long distances from San Diego to, let's say, Green Valley in LA. So we know that human-assisted dispersal is the main problem here, and that's with firewood. And that's, kind of the perfect storm. You have an oak dying, firewood is really valuable. The person who had the oak dying doesn't know what to do with the wood, and it's easy to sell. Here's free wood, or here's wood for cheap. And it's really hard if you are driving and say, hey, wood for cheap, and it's great wood. I'm gonna take it to my cabin up in the mountains, and that's what happened in some cases. So really focusing on not moving firewood, you see, I'm gonna kind of talk about that a lot. So now, when we think about an integrated pest management framework for this pest, I want you to think a little bit more broadly than where we usually think. This is kind of a complicated graph, but I'm going to show you a few things that I want to focus on. One of them is you need your research and your outreach together with your IPM, right? We're talking about your pest management strategies, but also communication with the other agencies, communication with the public. Very, very important to have a successful management program. And within the different, like the economical viability, of course, something you need to consider, the environmental safety, and the social receptibility. So when you're doing something, it's very important to have some sort of outreach campaign together with it, because you need to have the public on your side. You might be removing trees, you might be treating trees, so you really have to include that within your management plan. So we talk about working with researchers and experts and communicating. So we have our pest management plan that has monitoring root trees, so removing root trees or amplified trees, managing infested wood, protecting other trees, reforestation, but also very important communication with the public, right? When are you doing things and why are you doing it? Why are people removing my host. Make sure you engage and educate the community if you have any chances of having events and explain what GSOP is and why are you doing what you're doing. That's great. And of course, we always work on prevention. So every time we talk to the community, we're going to say, what are we going to tell them to do? And say, what can I do about GSOP? We can think of something. Don't move firewood. Yeah, exactly. So don't move firewood. I already talked about outreach to communities. We use master gardeners as a way to do a lot of our outreach, but any way you can let the public know what you're doing and why it's important. And then it's very important to have a monitoring plan, right? And for that, it's good to involve all stakeholders in the monitoring. You want the neighbors to be looking at the trees as well. You want, if you have volunteers, you want them involved as well. And they have to have proper training. This is regarding volunteers and monitoring. Stay tuned because we are planning on a GSOP Blitz in June. So if you do manage volunteers and you want to train them to look for GSOP, just let us know because we're planning on a GSOP Blitz. All right. Now, how am I doing on time? I found GSOP. So now what do I do? So the first thing you're gonna do is document what you see, right? You need to take pictures. That's the most important thing. What kind of pictures? Well, we want the exit hole. I don't know why I keep saying that. I'm sorry. You want the exit holes, right? But with something for scale. Now, look at your finger. Is your finger the same size as mine? Let's see. Oh, look at this. So, how big is this hole? Is it like 3 millimeters or is it like 5 millimeters, right? That's not a great idea. Better idea, use something for scale. Or even better idea, use a little ruler for scale, right? And these are cards that we do have them. And if you don't have one yet, I recommend you to get one from us. And if we don't have them out yet, Randall has them in the back of the room. And for the people online, if you don't have cards and you want cards, just contact us and we can send them to you. All right, you found it, you took your picture. Please take your picture and don't go at it with a chisel because unless you know exactly what you're doing, because sometimes, yes, it makes sense to just remove back ever so slightly the bark because the rugosities in the bark might obscure that T-shaped exit hole. But really, if you haven't done that yet, if you want confirmation, just don't touch it, right? I've seen some people were saying, I think I have GSOP. They sent me some pictures. I'm like, I think you might have GSOP. Let me go take a look. And we went to take a look. They had chisel each of the holes that they found trying to find the gallery underneath. And they got rid of all the evidence. So now we don't know. So if you want confirmation, don't chisel it, send the picture, and wait if you want someone to go check it out. Wait for that to happen. So you're gonna be collecting site information, the species, the DBH, how many exit holes you have, where this happened. If you have that long coordinates, that's better. And then you can report a suspected infestation at gsop.org. That's our website. That's in the case that if you have any doubts, like I think this is GSOP, this is a new location, I don't know what, just go and report it. And you're gonna need all of this. You're gonna need the pictures, you're gonna need all of this information in order to report it. Now, if you're monitoring for management, right, we want to survey once a year, ideally during fall and winter, and we want to record how many exit holes we find every time. And ideally, how many new exit holes we find if you're just gonna go year after year. Ideally, what we do in order to distinguish old exit holes from new exit holes is we mark them. Excuse me for my voice, I'm trying my best today. We mark them with a crayon or with a paint marker. That also helps to count, because if you have a tree and you're counting how many holes, and then you're like, did I count this one or not? But if you put a dot next to it, you already know you counted it. So, and then we're going to assess how the tree is doing based on that. So is it lightly infested? If it has less than 10 exit holes, it's moderately infested. It has from 10 to 25, and it's heavily infested if it has more than 25. Though usually this is the kind of trees that are considered an amplifier tree with more than 25. However, some people are starting to consider more than 10 as an amplifier tree. And that's something that we'll be looking into. Not today. We as the collective, we researchers. And then we'll report back. We're gonna take some notes on the crown rating, right? If it's how much dieback do you have, right? And so the idea is once the crown has more than 25% dieback, you're probably going to be looking into a tree that is not treatable. It's a tree that is probably already on the way out. And kind of similar with bark stains, depending on the number of bark stains, we can think how badly it is infested, right? Once you start having the stains and cracks that are horizontal, that tree is gone. But if you have more than 10 stains in the trunk, that's probably also a sign that that tree is severely stressed and it's probably on its way out as well. So how are we going to use the GSOP cycle and to time our management actions? So this is another version of the GSOP life cycle. We have our months up here from January to December. And as we say, so the adults are emerging between May and September, then they lay the eggs, then they feed throughout the summer and in the fall, and then they prepupate during the winter, pupate at the beginning of the spring, and that continues again. We could be trapping during the time that the adults are emerging. We used to use the prism traps, the purple prism traps, yes, without lure. And so because it doesn't have a lure and we don't have any lure for GSOP that we know of, they're not very effective. So we stopped doing the trapping. Plus, for a while at least, the manufacturer of the prism traps stopped making them. So I left it in there, but it's not something that at least I am currently doing. And then if you're thinking about applying contact insecticide, for example, you want to do it at the beginning of the emerging season, right? And if it's systemic insecticide application, you want to do it, maybe it says from February to April, but it just kind of depends on where you are. But you want to do it when the tree starts uptaking a little bit more, when it starts getting a little bit warmer. Mechanical grinding, and we'll see when we do that, but you can do it all year round, or why we do that, better say. So surveys, better do it after the emergent season, so you capture everything. And based on this, when would it be safest to transport any kind of infested wood to a management facility? Who can tell me? In the summer? Winter time. Oh, hi, Rachel. In the winter, exactly. We want to do it when the beetles are not emerging, right? I mean, if you do have to remove a tree, you can just do it anytime. But if you really have to move and you can choose, you'd rather do that during the winter when you can survey as well, so that there's less chance that the beetles are going to escape from that wood that you're moving to a management facility. And I said consider treating earlier in lower elevation areas, because this graph has been done with high elevation data. So it's a little bit warmer earlier here in lower elevation areas. And then my colleague Jolene Tam has been observing that actually it seems like the growing season, the growing season of the beetle seems to be extended. She kept finding adults up to November, larval feeding almost all year round. So it's something to consider, and it's something that we're going to be looking into a little bit more. Because if the emergent season is getting a little bit spread out, we might have to consider starting treating maybe twice at the beginning and in the middle of the emergent season. But that's something that we still need to figure out if that makes sense or not. Now, so for the things that you can do, if you have an amplifier tree, that means a heavily infested tree. These trees contain about 60 to 90% of the whole GSOP population in an area. So you want to get rid of them as soon as possible, right? Now, when you remove a tree like that, okay, hold a second. Why do you want to remove this tree? Stop the breeding? Yeah. So these usually are about 10% of the infested trees fall within the category of the fire tree. And they contain 60 to 90% of the beetles in an area. You remove those, you're removing 60 to 90% of the beetles in an area, right? And then you're doing active management. You're removing one of the sides of the disease triangle from IPM. You're removing the pest and you're lowering the pest pressure over there. Let me see how we're doing time. Okay, I gotta wrap up soon. You still have some beetles in that bark, so what you're gonna do with that wood, you can screen it. If you want to use it as firewood, you need to leave it screened for 2 years before you can move it. So not a very convenient way. You can debark it and use it as firewood. That's safe. You can use a chainsaw. You can use a wood splitter. There's a few ways you can do this, but it's pretty costly and labor intensive. Yeah, you can use, there's an attachment for your chainsaw that you can use for debarking. But then the easiest way is to chip the wood to less than 3 inches. And that way you're sure that all the beetles in that wood get killed. And then for high value oaks, you could potentially consider a chemical treatment. So you have to make sure that GSOP is in the area, that is a susceptible species, and that is a healthy and vigorous tree. We're not going to be treating a tree that is on the way out. And for options for that, we have contact insecticides that you will be applying to the trunk and the higher branches, the branches that are bigger than 8 inches in diameter. And, this one can have drift that could be dangerous for aquatic organisms. So you cannot do this spray next to bodies of water. And so in those cases, you're left with systemic insecticides as an option that usually goes as a trunk injection. But you have to make a hole. We've been seeing here the holes that you make, the drill for making the trunk injections. In some areas, they don't heal very well, even though we spray those holes with antifungal like a biological antifungal, but they're still not doing so great. So it is an option, especially if you have a body of water next by, but it's something to consider that sometimes it doesn't work as well. And then lastly, there's potential for biological control. We have these 3 species of wasp found here, parasitizing GSOP, but we need more research to figure out if it's possible. We know that Balca indica has some impact on emerald ash borer, another Agrilos species, but their impact is only 3%. They kill 3% of the population. So it is to be seen if it will work for GSOP and how good it will work. All right, last thing, GSOP.org. Don't forget, this is our website. Please come visit. You can report any GSOP sightings in here, and you can sign to our newsletter. So you get next time we have a training or something, you get an email. All right, thank you very much. And I'm not sure if we have any time for questions, but you can find me after if not. We use the Purple Prism traps that are sticky traps, are kind of sticky trap, and they do work sometimes. So they catch beetles, but it's basically when the beetles are flying, there's an idea that maybe they are attracted to the dark color, but they have nothing that lures them to that trap. So it's kind of sheer luck that you will get them, and they're not very reliable. You can use them, but if you don't do visual inspections in the area, if you don't catch anything, I wouldn't be very confident that you don't have GSOP.
And especially wood, does the solarization like plastic wrap work or no?
What you're doing is kind of wrapping it all, and it's not about killing the beetles with the heat. It's kind of like they will emerge and die of starvation. So you have to make sure it's super tight, and the plastic wrap gets broken very easily. Yeah, and you have to be contained for 2 years. So it's really hard to have plastic wrap in good condition for 2 years. Yes.
And also birds will jump on the plastic wrap and rip it up to try to get bugs on the inside. Longhorn beetles, round heads will come out and chew huge holes through it. So it's very difficult.
I'd like to make a request for the in-person speakers. When you receive a question from your live audience, will you kindly repeat it for the attendees online?
Covering with tarp will prevent the GSOP from dispersing with firewood. It will prevent it from dispersing on transit, but once you have the firewood somewhere, they will still come out of that firewood, right? Even if you were just planning on burning it that day, you still have a chance that the beetles are going to emerge at that time. So really, moving firewood is discouraged with tarp or without. Does that answer your question?
Thank you.
Hi, everyone. So I'm going to talk to you about, I'm going to give you a whole slew of information about different oak diseases in California. And so I'm just going to get started and keep track of time for me because I'm not sure. Well, it's better stop. So many bugs. Okay. All right. So just to give you a little bit of a background, oaks are foundational species in California and are important elements of both the natural and man-made landscape along, throughout the state. It's a prized tree is a symbol of natural beauty, as well as important parts of, preserve, as part of, supporting wildlife, or riparian and forest communities, and contributing to landscape resilience, following fire, and providing habitat and precision tree for numerous wildlife species. The history of oaks in California is long and storied and have had a place in California culture as well. For California Indians, oaks were perhaps single most important species, all of the species in general. They actively and skillfully managed these trees and the habitats to increase yields and sustain production of acorns to use for a variety of purposes, which goes back to 9000 BC. So unfortunately, oaks have experienced mortality since the mid-1990s. In Northern California, this mortality is largely attributed to sudden oak death, which is caused by this single invasive multi-host pathogen, Phytophthora ramorum. In the early 2000s, I was actually involved with a lot of the early studies on trying to understand kind of how this disease was spreading and moving throughout the state. So we're going to talk about this, but, in Southern California, oak decline is also attributed to gold spot oak borer and all of these other messy things that I just beautifully talked about. But there's other things that are involved, including pathogens and throughout the state that we're going to talk about today. So I'm going to talk about different fungi, different bacteria, and other things that are involved in the decline of oaks throughout the state, in addition to sudden oak death. So starting out with sudden oak death. So in the mid-1990s, there were hikers that were bopping around Marin County, and they were noticing this unexplained tan oak mortality that was going on in their neighborhoods that they couldn't figure out what was going on. This was then later noticed in their coast live oaks as well, and it was kind of integrated in this urban wildland interface. And it got coined this term while they were trying to figure out what was the cause of this unexplained mortality. The term sudden oak death got coined to try to explain what was going on, because what was happening was there was what appeared to be this rapid, all of a sudden, like the canopy would just turn brown. And what we found out later was this mortality would take maybe anywhere between 1 to 5, 6 years for the tree to actually decline. It actually looked like it was appearing much more rapidly than that, because it was kind of at the end of its growth, right? So in the summer of 2000, the cause of the mortality was attributed to this beautiful creature that, unfortunately, it is a beautiful creature. This is Phytophthora ramorum. It is an oomycete, which is actually a water mold. So it looks like a fungus and it behaves like a fungus, but it is actually not a fungus. It is more closely related to brown algae. And it makes these sporangia that contain these spores, called zoospores, inside of it. And these zoospores can actually swim through water, which is very unique. Fungi aren't able to do that. And so these are dispersal structures that allow it to basically move through water. And then it also produces these chlamydospores. And these chlamydospores are these melanized spores that have this really thick cell wall. And these are survival structures. And these allow the spores to basically over summer, if you will. So they can survive dry conditions when there's not ample water. And it allows them to, survive those harsh conditions until the conditions are good enough for them to germinate. At the time when this was discovered, they found that it was an unknown species of Phytophthora at the time, and they actually then linked it to this Phytophthora species that was causing blight in nurseries in Europe. And so even though it was causing mortality on oak trees in California, they actually found it was causing blight on foliar blights on nursery plants in Europe. And so they knew that it was not only causing mortality on trees and affecting the bowl of trees, but it was also causing dieback and leaf blight on nursery plants in Europe. And so they basically figured that it was probably coming in through nursery plants and potentially escaping into the forest. So this is basically the map of the distribution of sudden oak death. It's, currently found in 14 counties, and it's also in Southern Oregon as well. There is also, they've developed a risk map of sudden oak death. This is based on the degree of risk is based on climate, host range, and proximity to nursery operations. So it does have high potential to drop down into Southern California within the coast range. There is moderate risk, up through the Central Valley as well. But its temperature tends to be optimal around 70 or 68 degrees. That's where it tends to thrive. And that's why it's been so tightly restricted to kind of strange. The disease symptoms caused by Phytophthora they differ depending on the host. So it primarily affects, as far as the things that it kills, it affects members of the oak family. So true oaks, so members in the Fagaceae, the true oaks, so members in the Quercus genus, as well as tan oaks. So this has changed, so it's Carpus densiforus, as well as chestnut in Europe and beech in Europe as well. So this is just looking at, so if you were to see these symptoms in Southern California, what would you think that this was? Drought, GSOB, potentially, right? So location matters, context matters, and everything, right? So if you're looking at this whole crown and you're seeing this massive dieback in the crown, a lot of people think, oh my gosh, it just could be GSOB. Or, people see a bleeding canker and they think stomach oak death, right? You saw bleeding cankers on the tree associated with GSOB as well. People see bleeding cankers sometimes, and they when they're in Northern California, and they think sudden oak death, and then they come down to Southern California and they see bleeding cankers and they think, oh my gosh, sudden oak death is in Southern California now. So it's really important to understand that just because you see a bleeding canker on a tree does not mean that it's attributed to one single causal organism. It's really, really important that you have to either test it in a lab or look more closely at what is going on on the particular tree itself. In particular, for sudden oak death, the bleeding or oozing is on the bark. It's not associated with cracks in the bark or insect holes. So it's just kind of oozing and seeping through. It can be it's usually on the lower 6 feet of the tree or the trunks, but I've seen it, make its way all the way up the bowl. When you peel back the outer bark, you see what's called a canker, and the canker is restricted to the phloem. So if you're looking at the inner bark of the tree itself and you're peeling back, you see what's called this necrotic tissue here. So this is the dead tissue here, and this pink here is the healthy tissue. And what's interesting is then there's this zone line here. The zone line is what's separating that dead and that live tissue. And that zone line is actually the host responding. It's actually laying down pigment called their anthocyanins. It's laying down pigment, and that pigment is an attempt to kind of wall off the pathogen from trying to move and advance through the plant tissue. This white here, sometimes you might see this white streaking. That white streaking is callicing that's being produced by the host itself. That callicin is, again, another response by the host, and that's trying to wall off the plant from allowing the pathogen to advance through the host tissue. But this canker is something that you see with some milk death, phloem canker. However, there's a lot of fungi that produce these phloem cankers as well. So again, just because you see a bleeding canker and just because you see a phloem canker, it doesn't necessarily mean it's one causal organism, particular causal organism. Oftentimes, you have to take this back to the lab to get it tested, to get in the diagnostic lab to get tested. This is a bleeding canker that's caused by something else. Does anybody recognize this? I think gave it away. So this is caused by armillaria root rot. Okay. So this is actually caused by a fungus itself. The fungus, it's a fungus that produces these beautiful white mycelial bands underneath the bark, at the inner bark, and sometimes like at that layer of the cambium as well. Okay. And sometimes you'll see what's called rhizomorphs. They look these black, stringy things that look like roots, but they're actually being produced by the fungus itself. This picture doesn't show them very well. But anyway, when you look at the outer bark, you see bleeding cankers. And so looking underneath, you see something completely different. This is showing a bleeding canker. However, it's linked to a little hole there. Okay. There's a boring insect in there. There's a little piece of larvae. I'm not sure which boring it is, but if you go into the inner bark, there's a that's right. This is a canker also caused by a canker rot pathogen. So again, it's causing necrosis on the same part of the tissue, but it is not the same species. So it's really just driving home, it's really important to take samples and get the lead out of lab. So we've got many other common diseases, many other things that can cause diseases on oaks. Wetwood, you get this long bleeding sap spillage, and it stinks. It smells really bad. That is, wetwood is caused by a bacterium. It doesn't smell good, and it's usually associated with cracks. You got boreal insects, mechanical injury, and lots of fungi that we're going to talk about as well. But one of the things that's important to drive home is that there's two sets of symptoms that are caused by bipolar morm. So we've covered the oaks, but there's also foliar blight. So this is on non-oaks. You get spots and blotches on leaves. You get shoot dieback. You can kill some juvenile plants, occasionally mature plants, but for the most part, it doesn't kill these plants. For the most part, you just get these foliar blights. So one important host, I'm just going to give one example, is California Bay laurel, where you get just these little tip blights here. So you get this necrosis and you get this little zone line here. And what's happening is you get what we see here on the tips is, well, if you look under a microscope, you see the sporangia just sitting there at the tips of the leaves. And we call these hosts reservoir species. And they're reservoir species because they're harboring inoculum. They're not killing the plant, but the plant itself is just serving as a reservoir of inoculum. So then, for the oak trees die and they're basically a dead end host. But with something like California Bay Laurel, it serves as this perpetual source of inoculum in the forest. So when it rains, you just constantly get, splash of sporangia that rains onto the oaks, and you just get spores raining on into the forest system, and it never leaves the system. So in terms of management, what do you think practitioners are doing in a forest setting? What's one of the biggest ways that people are managing this? Yeah.
Is it viable on the reams that are falling down into the ground as well?
They had, yes, we have recovered it from leaf litter. It's not as viable year round, but during the winter, it is viable.
We're spraying with like phosphoric acid on 1.2.
Good question. They are spraying that's a good question. I don't know about the foliar trees, but the foliar pests, but for key trees, they are doing injections of oats. So that is one tree.
Going back to the injection that works for a basal activation, sufficient as well, or you can push them through phosphorus and pretty.
That is something that they have applied, yeah. But one of the biggest things that they have done is for like management, where you can't really do that on a large scale, like in a forest setting, is they've actually done bay tree removal. So they've done selective removal in bay trees. So to reduce the source of inoculum. I don't know if somebody said that back there.
Yeah, we did. We were going.
I just heard like so.
So
When you get a question. Oh yeah. Sorry, Zoom people. Yeah. Yes. So selective bay tree removal. So some people in the back got it. I just didn't hear. But yeah, selective bay tree removal, reducing the source of inoculum in order to preserve those oaks in the forest in one way to manage the disease.
I feel like, proximity guidelines, tolerance for the middles to bay trees and forage.
That's a good, I don't have it. So the question was, are there proximity guidelines? Like, do you remove a bay tree a certain number of feet from an oak? And the truth is I don't have a good answer for that. I don't know. But I just wanted to throw this out there. This pathogen has a very broad host range. So you can see how it is a very messy thing to manage because, and this is why it can survive so readily in a forest, and why it's really difficult to objectives.
If you were going to use any of these species for like a sentinel tree for monitoring purposes, which species would you pick?
If you were going to monitor these trees for sentinel tree purposes, which tree species would you pick? That's a great question. What do you guys think? California Bay? Yeah, that would be a good bay tree.
That would be one.
Depends on the context too. There's not a lot of bay tree in Southern California. So, I mean, you could potentially use it as a bait tree, right? But put out a few bait trees like in pots or something like that. But yeah.
Okay. So moving on, we're going to talk about acute oak decline now. So this is something that
is popping up and emerging. So what is acute oak decline? So again, we've got these big bleeding cankers. And so bleeding cankers is the thing. I think I'm going to dress up as a bleeding canker for Halloween. So acute oak decline was first identified and characterized on native oak species in the UK in 2009. It's a complex disease of oak with no single cause. And it was first characterized by this researcher, Sandra Denman, in the UK. She's a project lead, this group called Stop the Bacteria. The trees in the UK, we haven't seen this happen in California, but in the UK, they die within 3 to 6 years from the onset of symptoms. And basically what they have is, Sorry. Basically, what they've established is that acute oak decline, or AOD, is caused by this combination of environmental stress, beetles, and bacteria. So environmental stress, particularly drought, weakens the tree, and then you get beetle colonization. The beetles that they have identified that are associated with this are a bupressid beetle. So does anybody know what bupressid beetles we might have in California? That's but some would say metallic wood boring beetle. Yeah. The one with spots? Well, not spots. Gold spotted oak borer. Yes, good. Good, Hannah. Good. Gold spotted oak borer. Exactly. So it's a bupressid beetle, okay, that they have in the UK. And it's this combination of these bacteria, Renaria, Gibsiella, Renalia, and Ladsodella. Okay. So it's this combination of these bacteria. It's not one single species, but a combination of these bacteria that interact with these beetles after they've been predisposed to stress. So this is just showing the distribution of those reports in the UK. And there is basically this significant correlation between this AOD occurrence and warm areas and dry areas that are prone to drought. They've also seen a correlation with high nitrogen and low dry sulfur as well. So now I'm going to kind of take us back to 2008 in San Diego County, when I was working at UC Riverside, and we were getting calls of unexplained mortality of coast live oak that was associated with bleeding cankers. And many people in the public were concerned that it was because of sudden oak death. And that's why we were getting calls in the lab that I was working, and that's how kind of I got involved. So we were going and we were looking at these trees. And what we found was these larvae instead in the cambia layer. And so now we know that these bleeding cankers were caused by gold spotted oak borer. At the same time, we saw these cankers. When you peel back that outer bark layer, we saw these larvae, but then you saw these flown cankers as well. That one could say, oh, that might be sudden oak death too. But we instead found this fungus growing out of them. This fungus was Diplodia corticula in the Bacchus fariaceae. So this is a family of canker pathogens. And there were other locations with oak mortality where gold spotted oak borer did not occur. And we were recovering Diplodia corticula and from the trunk as well as from branch on declining oaks. And we knew at the time that Diplodia corticula was a known contributor to cork oak decline in Europe. And so in the greenhouse, we did pathogenicity tests and we inoculated saplings. And actually, within 4 weeks, Diplodia actually killed the trees. It moved through the xylem in advance of the phloem, and it actually went into the roots of these saplings, which is not normal. It's not something that we actually see for this class of fungi go down into the root system like that and to move so quickly through. And it reproduced these symptoms of bleeding, and it was just very rapid and aggressive. And we were also fortunate enough to be able to inoculate experimentally in the field, of course, with permission, and reproduce these symptoms in the field as well. And so we found corticula to be pathogenic on coast live oak, and we recovered it from gold spotted oak borer infested and non-infested sites in association with different plant parts. But the other question remained, are there differences in mortality between these different sites, and what factors drive pathogen and exotic distributions? And so we set up a study where we put in monitoring plots in GSI infested and non-infested sites to look at mortality throughout Southern California. And we sampled the trunk and we sampled branches, and we basically looked at all the different fungi from these different things. And so this is basically just looking at the proportion of dead trees from our plots that are GSOB infested, that were GSOB recently infested, and that were not infested. Basically, we found that at least between the non-infested and the infested and the non-infested, that there wasn't any difference in mortality between those sites. And this graph is showing basically annual precipitation and the proportion of trees with, should we all stop killing there? Yes. No. I'm sorry. Let's fix that up to the brain. Okay. So annual precipitation and the proportion of trees that have gold-spotted oak borer and Diplodia corticula. And what's interesting here is that in the infested, recently infested, and non-infested sites. And what's interesting is that we found there's obviously, so there's more corticula in the GSOB non-infested sites, and that's linked to annual precipitation. So there is this kind of pattern across Southern California where decline is associated with different disease-causing organisms. GSOB is much more devastating than Diplodia corticula, but there is this link between decline and Diplodia corticula, and it's linked to these, and it's linked to these kind of nuanced differences in precipitation throughout the region. Yeah. That question. So areas with more precipitation had more GSOB. Yeah. And the areas with less precipitation had more. Yes. The particular, yeah. Is that because drought stress might be a contributing factor? It could be, and it could just be more like, it could just be kind of a nuanced thing though too, because in our greenhouse studies, those were well-watered seedlings, and the pathogen moved really aggressively through those plants and killed them quickly. And so they weren't drought stressed when they died. So it's the trees that, in these locations could be drought stressed, or it could just be, all of these trees could be drought stressed, but it's just, or they could just be, expressing nuanced levels of differences in participation. Okay. And then we also found other fungi as well. So that's the main conclusion. Coast live oak mortality is linked to GSOB and corticula with non-overlapping distribution, which could be incited by long-term drought. The GSOB and corticula occurrences on a finer scale are linked to annual precipitation. More precipitation in GSOB, less precipitation in corticula infested sites. Yeah. So I wanted to introduce that because then around the same time, this Gibsiella greggii, okay, so we're coming back to this acute oak decline, okay? This bacteria pops up on a single black oak tree in 2010. And this pops up on a tree that happened to be on a GSOB infested tree, okay? So like in all of these surveys and everything like that, this does pop up. But so that pops up, and then all of a sudden in the last few years, there's been this emergence of these trees now where we're getting more records of what's called this acute oak decline, where we're seeing these symptoms of these trees that have bleeding cankers, and we're recovering these bacteria that are of the same, that are similar species like the ones in the UK. But what's really strange is, so we have some that are popping up in Northern California, and we have a bunch that are popping up in Southern California. And what's weird is that all but one of the locations are gold spotted or borer free. So the story is very different. It's very puzzling. So we don't know what is going on. The story in the UK is very tightly linked to this bupressic beetle, but we're getting these records of these trees popping up in California, but they're not tied to GSOB. They're happening coincidentally, in some rare cases, but not all of these cases. So the symptoms include crown thinning. They include these weeping patches on the tree trunks. So again, they look very much like gold-spotted ochre, but what would you see if it was gold-spotted ochre? V-shaped enzymals. Thank you. V-shaped exit points. Yes. That you would see, in addition to when you come up closer to the tree, in addition to these long, like just big weeping stains, you see longitudinal cracks in the bark and a gap between the bark plates that kind of widens. And then if you move into the cambium, and basically you get these phloem cankers and that kind of extend into the cambium layer as well. So again, you go in, here's the this is kind of getting into this is actually all callousing in here, but you get this necrosis here, dead tissue, and the healthy tissue, separating dead and healthy tissue. And then just some more pictures where we've recovered basically pictures of these symptomatic trees. All of these are examples where we've recovered these bacteria species from these symptoms. We haven't gotten fungi, we haven't gotten the cortical, we haven't gotten Phytophthora remora. We've gotten these bacteria that are attributed to acute oak decline, like what's causing acute oak decline like that in the UK, but they're not overlapping with any kind of beetle at all. So we're trying to put the puzzle pieces together here. So we've confirmed Gypsiella greivii, which is closely related to the Ciella species that's in the UK. It's not the same species, it's closely related. Renaria goodwinii and Renaria victoria. Oh, yes. Are these bacteria now stored in the soil? So the question is, do these bacteria occur in the soil? We've tried to recover the bacteria from the soil, and we haven't done a good job of trying to get them yet from the soil. That's one of the questions that we have is what is the life cycle? But the first thing that we need to do is confirm pathogenicity on the trees. So we're consistently recovering these bacteria from the trees, but just because you recover an organism from a tree or any plant, that doesn't necessarily mean that it's causing those symptoms. And so the important thing that we have to do is do the pathogenicity tests. And that is what we are working on right now. And once we get that confirmed, so we're in the process of doing that in the field and in the greenhouse. Once we confirm pathogenicity, where we can inoculate the healthy plants with the bacteria and show that it can reproduce the symptoms, that it's reproducing that necrosis, and then that we can take that necrosis and recover the bacteria from the necrotic tissues, and that completes the pathogenicity test. Once we can do that, then we can say, okay, now here's our questions. What's its life cycle? How is it behaving in a landscape, across the landscape? What is its incidence across the landscape? What are all of the different ecological questions that we have? So after the pathogenicity test, we're going to conduct surveys of oak woodlands to determine occurrence and severity, determine factors controlling the disease life cycle in California, including its behavior in the soil, understanding how it reproduces, spreads, and survives, and determine how it interacts with other fungi, and then work on developing best management practices.
You don't need to answer my question.
Okay, great. Okay, great. So if you want to know more, our colleague Jolene made this lovely flyer. And so this is a living document that you can get. It's constantly being updated. So if you want updates, you can go there. I've got one more fun thing to talk about. Do I have time? Yeah, because I That mistaken and I finished them all. Okay, perfect. So we'll talk about foamy bark canker. Can we do a couple questions? Absolutely. Yeah. Can you describe the visual symptoms of Phytophthora cinnamoni versus Phytophthora ramorum? And is P. cinnamoni more treatable? Phytophthora cinnamoni, that's a great question. They're going to be very similar to Phytophthora ramorum. So Phytophthora cinnamoni is another Phytophthora species that can affect oaks. And it's a soil-borne pathogen in contrast to Phytophthora ramorum, which what they call an aerial Phytophthora, because Phytophthora ramorum is not soil-borne. It affects above ground. It affects like the bulb and it spreads through the above ground tissues, whereas Phytophthora cinnamoni spreads through the roots and it's soil-borne. So with cinnamoni, you're going to see similar symptoms of bleeding cankers. They're going to probably be lower on the bulb because they are soil-borne. So they're not going to spread. They're not going to be found like as high up on the bulb as something like Phytophthora ramorum. Phytophthora cinnamoni is not going to be causing any foliar blight. Phytophthora cinnamoni also has a broad host range, but it's not going to be causing tip blights on other tree species. The main thing that you want to look for is kind of full crown dieback that's very uniform and kind of patches of disease centers because it's spreading from root to root. So you're gonna see little patches of trees like dying together, like Dublin woolings. They're all dying together in patches. As opposed to stud and oak death, it's gonna be more, and you're gonna see disease centers, whereas with stud and oak death, it's more patchy. Dead tree here, dead tree here, dead tree here, dead tree here, intermixed with live trees. That makes sense. As far as treatment, the treatments are the same. There's a follow-up. Are most labs equally trained and outfitted to determine the species of Phytophthora? So the problem is not all labs have the permits to test for Phytophthora ramorum. So CDFA has the permits to test for Phytophthora ramorum. Dave Rizzo's lab has the permits to test for Phytophthora ramorum, and Matteo Garbonato's lab has the permits to test for Phytophthora ramorum, and that's pretty much it. Yeah.
Just to be fair though, if you're a private lab and you're isolated by, you don't know it's Phytophthora.
You don't know. Correct.
Until you isolate.
Yeah. And then, it's not, if you do it unintentionally, it's not like you're breaking the law. Yeah. Okay. All right. So let's talk about one more disease. It's more of a pest pathogen associated kind of complex thing that we're still trying to understand. So a little bit of background. We saw a decline of coast live oak in California. This has been observed since late 2012 throughout urban landscapes and private lands in LA, Orange, Riverside, Santa Barbara, Ventura, and even in Monterey counties, and even more kind of throughout the Bay Area as well. So again, you see this dead tree and it could be anything. Right? What's that? It could be anything. It could be the golden spotted oak borer. It could be sudden death. It could be horticola. It could be anything. So going into the canopy, just like these huge trees, just suddenly dying. And when you look up closely, we saw these trees that had these kind of round stains going all the way up the bowl, up into the primary branches. Does this look similar to anything that you've seen before?
On walnut?
Yeah, similar to thousand canker. That's a good observation, actually. Someone said thousand cankers on that you see on walnut. Really good observations. Anything else?
Some versus shop.
Shovel borer. Yeah, liking that we're always making your shacklebore. The lichens that you think are shacklebore. Yeah. So yeah, it kind of looks like shacklebore, right? The kind of the small round stains that are going all the way up the bulb. Yeah. So very, very similar. But then there's this, it's hard to see here, but then we saw this kind of white foam coming out. And we were seeing this in a lot of places, and then just the whole tree just kind of like dies. And what we were seeing actually when we were looking closer at these trees, and we'll look at this a little bit more closely, we saw tiny, tiny entry holes. Okay. So the entry holes are actually much smaller, that you could barely see them. It's like the size of the tip of a sewing needle. You know, remember, what's the size of the shacklebore entry hole? The tip of a 110, right? So these are like the tip of a sewing needle pen. I don't know. But it's very, very tiny. Okay. So this is the Western oak bark beetle. And the Western oak bark beetle is a native beetle to California. It is Pseudopterotheris pubescens. We identified it morphologically, and we identified it based on molecular methods as well. And what's interesting is that this little guy is never been associated with a fungus. It's a secondary insect. It tends to affect stress trees, and it's not really, it's not necessarily a primary anything like that. But what was odd was that we were consistently finding this fungus, Geosmithia, within the galleries of this bark beetle. So this is a bark beetle, and this Geosmithia species turns out to be probably native to California as well.
Okay.
So this is where we've been finding it throughout the state, all over California. And it exudes this, it's pretty, like on these live trees, when it attacks, it exudes this foam. And you can see the tiny beetle, it's a host response, right? So that it's pushing the beetle out. So here's this foam here, and it's pushing the beetle. It's pushing the beetle out. So that's where it got the name foam bark canker. And here's the fungus that we found in association with the galleries. Again, this is not kind of something that we've seen before. So it's something that that we still don't really understand. It's never been associated with a fungus. This is a new thing. I think we even tried chopping its head off, because beetles tend to have what's called special components in their mouth called mycangia, where they carry spores to transmit from a fruiting tree. And so we chopped off its head to see if we could get this fungus out of mycangia. But I don't even think that they had
I don't know. And they could be anywhere. They could be on the
they could be, yeah.
months venture. They could be all over.
They could be anywhere. So it's not necessary. So the association between beetles and fungi can be very tight, like an obligate mutualism, like you see shacklebore, or there can be very loose association where they're just picking it up, accidentally, which could be the case here. This new thing that is happening, but it's happening pretty often throughout the state and very recently. So it's kind of an interesting thing. And just one more slide that I'm going to show you so that you can see right here. So it's happening in the bark right here. So it's a bark beetle, and you can see the fungus is basically colonizing the clone tissue, causing cancers and girding the tree. And that's where we get that mortality.
And I found it this far in Cascades and Black. And Chris has found it up on the coast.
Interesting.
So I know that map was the initial one.
Yeah.
First found.
Yeah.
It's been popping up all over.
Okay. So that's pretty much it as for foam bark canker. So it's an interesting pest pathogen complex
And it also does seem to be limited to stress trees.
Yeah.
Like this, I'll go and visit the site, it'll be up on rise, so it's, maybe a limiting factor. And it'll all be in like 5 or 10 trees all in one little spot.
Angie sub-infested trees. Angie sub-infested tree, yes. Okay. Interesting. So there you go. But kind of an interesting story.
So anyway,
So that's all I got for you today. If you're interested, there is an oak woodlands disease management manual that we developed back in the day, but it still stands pretty good and it's available online. So you can actually Google it. There's a PDF version of it online if you want to check it out. And thank you very much for your time and for your attention.
Shannon, we have a couple of thank you for your presentation
What's that?
in the chat that haven't been answered yet.
Having questions.
We have two. What is the difficulty of treating sudden oak death in coastal settings that have regular fog?
Regular fog. So because of moisture condition, because Phytophthora is wild and it requires moist and it thrives on moist conditions. So when you have, perpetual moisture in a system, it's just going to continuously thrive in that system. So it's really, really difficult to manage.
Thank you. And the one remaining question is regarding the multiple oak pests and fungi that we're experiencing. Should we not be planting oaks anymore?
Yeah, you should plant oaks.
You never know, some can be resistant.
Some can be resistant. Exactly. Yeah.
So with going towards the phosphoric acid for the problem, is it a one-time treatment or is it an annual treatment? And going more towards that fog question, that if the conditions are always persistent, is it something that you're doing on an annual basis? As opposed to if you're in dry lands, you can probably get away with one putting with the renewable oak trees.
So the question was about phosphoric acid treatments, and depending on the conditions, whether you're in a foggy area versus in a dry area, and how often you will be applying those treatments. And I think the main thing is, I think really the answer is about context and then where you're managing and what you're managing for. So are you managing in a wetland forest versus an urban forest versus, a high value tree in this situation? So phosphoric acid treatments aren't really going to be applicable to a wildland forest setting. And so that's where you're going to do more active management with bay tree removal and things like that. And with phosphoric acid, you're probably going to have to do those applications. The frequency of applications is going to depend on their environment.
Okay, so thank you. I'm going to be presenting on the Mediterranean oak borer. This is a new pest. It is an ambrosia beetle, like the invasive shothole borer. It's in the same tribe, so it has a lot in common. So if you know about them, you'll know a lot of it might seem like we do. So let's start with the critter itself. It's an invasive pest, native to Europe, Middle East, and North Africa and the Atlas Mountains. And I'll have a map in a little bit. In Napa, it's been around for at least 9 years. So it was first found in 2017, but the beetle was not identified. Only the fungus was taken because it was given to a mycologist and he went, a beetle, threw it out the window. But when we went back and looked where those were first collected, there were trees that have been dead for a long time. I think even 12 years is probably an underestimate. The females, if you go into the literature, it says 2.5 to like 3.6. I've measured 200 or 300 of them. 99% of them are 3.1 millimeters. So it has to do with where they come from. These come from a single point
population. So there's less variability in both their behavior and their size. The males, you'll almost never see them. They live way down in the deepest parts of the gallery where they mate with the females. On rare occasions in other species, they'll come crawling out fairly regularly, but I'll talk about why you rarely see them crawling out of the galleries. If anyone's interested in identifying themselves, there's 2 papers you'd need. This Rabaglia and Smith et al. From 2020 about the invasion in North America, and that just has a few couplets, which means you also have to get this Gomez 2018, but both are free online and can be found. If you're just sorting and you're just collected some beetles and it's about 3 millimeters long, and you think, well, maybe it's a Mediterranean oak borer, you look straight down on it, the butt is really straight across, where pretty much all the other ones that are in the Xyloboris, Xylobrini are going to be at least slightly pointy. That's an easy way. And then it's a disease complex, like the Eishab, also has Fusarium, just like the Eishab. But the Raphaelia montetii, it's been the most widespread detected fungus in Europe. It also seems to be the most widespread here. The Keith Escalem and Davis did the Koch's postulate, drilled little holes, put the various fungi in the hole, saw how much they spread, and it moves pretty slowly over like 100 days. It only went at 16 millimeters, 16 millimeters, 17 millimeters. So it moves fairly slowly. There are other things like laurel wilt, which has a yeast stage, and that'll zoom through the whole tree, through the xylem in very short order, a few months, where this does not appear to have yeast stage. It just goes slowly by hyphae through the wood. There are some others, but they don't seem to be so far as important, but it's a little bit understudied. And this insect, especially compared to Eishab and even GSOB, is very understudied. We've had very little funding. I've done a lot of it myself out of my own pocket and just begging for stuff from the Forest Service for traps and lures and stuff. So I unfortunately am not going to have as many recommendations on how to deal with this, and certainly not as solid of evidence, but there's a few things we have established. Okay, so he was first discovered in Calistoga. This is all the way at the north end of Napa Valley. It's in oak wood, probably came in oak packaging or something like that from wine country in Europe. So that was around 2018. The CBFA did delimitation as soon as, let's set out a grid, set up hundreds of traps in cooperation with the county ag commissioners in Sonoma and Napa and Lake counties. We detected it within a few months in all 3 counties, and by state statute, that's when we folded up the tents. It's established, no more funding. They just stopped paying attention to it. In conjunction mostly with Mike Jones with UCAM, we continue to trap. And this year I've got a bunch of cooperators to help me out. I've got traps down here and I've got traps up here. I'm going to hopefully get some more up here soon with some of the county ag people. We're also this year we've deployed temperature sensors, both external and internal probes in the trees, to see if we can figure out what is the trigger for the dispersal, because it has a kind of a wide window in the spring when they disperse. What's really important is going to be that xylem temperature. So it's now in 7 counties and presumably still spreading. And this is another one that, just like the gold-spotted oak borer, has a perfect biology for spread through firewood. Don't move firewood. Basically, in the fall, the trees die, but those trees are just packed full of females. Most of them are mated, and I'll get to it later. It doesn't matter if they're mated or not. They can still attack another tree, just one beetle. The trees get chopped up, they get moved around for firewood for the winter, and then they all come flooding out in the spring in the wood that wasn't burned. So the hosts are oaks in the white oak section, which the important ones, there's a few shrubby ones, but there's no evidence they attack them. Primarily, we found it on valley oak, less commonly on blue oak, but it certainly does attack blue oak, especially if there's a lot of valley oak around. It'll just go for all the blue oaks. And it took a long time. We had plots set up in Oregon oak, forests for like 3 or 4 years. Finally, there was a big fire, the glass fire that came through Napa. And then the next year that went through in the fall and the spring, we found attacks in Oregon oak, and then up in Oregon, they've got a separate introduction from a different part of Europe. And actually, there's very little genetic difference, but behaviorally, they do seem quite different up there. But they also only seem to attack lower in the tree, and I'll get to where they attack in California, and they only seem to attack very stressed out Oregon oaks there as well. So the native range is from here all the way over, even up into southern Scandinavia, all the way into Iran, and then in the Atlas Mountains up to Tunisia. There's what's called the same species, Xyloborus monographus, in the Iberian Peninsula, but the DNA is very different, 17% divergent, so it's likely that it's different. And from what I've seen in the literature, they, again, seem to be some behavioral differences. So there's quite a bit of literature here because they attack cork oak, which affects their wine industry, but there's some things about it that don't seem to apply. And then the other thing that is interesting is ours come from here in France. The Oregon ones come from Germany and the Rhine Valley. And then as you go further east, they seem to become less and less specific on oak. They start attacking a lot of other species. So in the literature, you'll also see probably up to 20 or 30 host species, but ours do not seem to attack anything else. So in their native range, they're pretty much limited to just dead or dying limbs. They'll be in the tree just for a few months because they attack it right at this kind of critical point where the moisture level is just enough for them to reproduce. By the time they're done with one generation, the moisture level's too low, so then they move to another tree. And in terms of research, most of the research is actually done for post-harvest because it ruins the value. And even there, it's a fairly low pest rating. It's the lowest, actually. So the range, dead or dying, usually one generation. Fifth tier, pest, least important. One paper that's looked in pretty good detail at the biology. It's in German. With the help of my brother who lived there for about 10 or 15 years, we've kind of done a pig German sort of translation of it. It's good enough to get the gist of what's going on. Very rarely 2 gens. And then what will happen is they'll come out in the March, April. These are the ones that have overwintered. They'll just do one generation. Then they'll come out with another peak in June or July. And then those are the ones that then feed over the summer to create that overwintering generation. Here in California, at least up in Northern California, there's really just one long singular, kind of like a 2-bump multi-peak. They get a cold spell right as they're starting their peak. Then they'll kind of like, oh, we're going to wait, and then they'll come out again. But they do not disperse again in July. They just continue to reproduce easily for 7 years. We've been tracking trees for 5 years. When we started tracking, they were already pretty well infested, and the decline is only like another 30% or so, so they could easily live another 5 years. So it's possible they could be in there for 10 years or 15 years even, but we'll see. We're going to keep tracking those plots. Oh, and I used to have slides for the plots. They also seem to, based on these plots, have the biggest effect on stressed out trees. So we've got like 6 plots. There's one that's right between the Napa Valley Wine Trail, the Calistoga City wastewater treatment plant on the other side. And then there's a ditch they made. In the winter, that ditch completely floods. So they've got heat stress in this summer, that water soggy stress in the winter, and those are by far declining the fastest of all the trees you see. So stress seems to be very important. Again, they seem to only really attack live trees. They have multiple overlapping generations. I've observed oviposition from early in the spring all the way into the first few weeks of October. I haven't looked later than that, but if it was really warm, I see no reason they wouldn't still be ovipositing in November. And then even in December, if it's warm enough, January, we had 80-degree days in January one year, and they were just pouring out of stumps and wood. So they don't really care that much. It does seem to be temperature driven, which is why we put out the temperature sensors. So this used to be one big confusing slide, and I redid it last night. Let's see how well this works. So here's a tree. It was attacked over on this side. I'll get to that a little bit more later. So basically, they come out of the tree in the spring, and each year that we've trapped, the end of May in 2020, early April in 2021, 2022, end of March, end of April 23, 24, early April again. So just highly variable year to year. And you can get 300, and that was like an average. One trap at 800 or 900 in one trap during a 1 week period. So the foundress goes in, starts excavating galleries, has these little pockets that feed the fungus, keep it pure at the base of the mandibles. So as soon as they start chewing, they're inoculating the tree, and that fungus starts to grow in the sapwood, starts to clog it up a bit. So let's say, they already covered that. So then from that point on, from March, whenever they attack, they start laying eggs. So you have that first foundress. If she's mated, she'll immediately start laying eggs. The fertilized eggs will be females. The unfertilized will be males. If it's an unfertilized female, she'll lose one generation, right? Because then she'll lay all males in that first generation. Then she'll mate with those. And then within a few weeks, well, a month to a month and a half, she'll start to produce females. And then depending on temperature and a lot of other factors, they'll just continue in the cycle. You can open up the tree and there'll be beetles in every stage, adult ones running around, all dark brown, ones just coming out of the pupa. There'll be eggs, every layer, at every stage of larva and pupae. So definitely overlapping generations. And, that's very much like honeybee. In this group, there's some that are called quasi-social, and they even have, a lot of the females will never leave. All they'll do is tend the larvae and the gardens. So they're not as social as honeybees. They don't have, females that are non-reproductive. But in some cases, there are females that won't even mate. They'll only take care of things in the colony. And then at some point, so why do they leave at different times during the year? So let's say it's August and all of a sudden the moisture content goes too low, their fungus will die, and then they'll kind of trickle out at that point. But at some point, they're all going to disperse, and it's usually the big peak in the spring. So this is a tree at a winery in Napa Valley. Been tracking this just visually for a few years. This is where they attacked. You can see it's similar to what Bea was showing, with the little clumps of leaves on her black oak. The arborists in Napa call it popcorn, but unfortunately seems like a lot of things can cause that. The beetles are all the way down on the back side of this bowl. It's all infested. There's none on this side, and the canopy looks a lot better on that side too. Unfortunately, to diagnose it, it means that for the first 5 years, all the beetles could be like on a branch up here. So unless you go up and climb, in fact, we climbed trees with buckets, taken the limbs down, and all we found was Pseudopodiopterus and stuff. And then a year or so later, the tree died and we took it apart and they were there, but we just couldn't find them. So it's very, very hard to diagnose. So symptoms of attack, the best thing, when we finally identified the beetle, it was October of 2019. There were a lot of branches falling that year. They were falling all over the place. Finally, some of the samples got to Sherry Smith with the Forest Service, the regional entomologist. She recognized that she didn't recognize it. She sent it to Bob Ravaglia in Washington, who identified it. Okay, finally, we found out in fall of 2019, we've got a new pest. And fortunately, if limbs do just crack off, which is actually fairly common, most ambrosia beetles, as they're chewing through the wood, they can sense each other. They can hear the little snips as they're biting through the fibers, and they'll actually kind of turn away from each other. These guys are different. They go toward each other, and they link them all up. In this way, like females from this foundress, original attacker, can then go in and mate with the other one. So that's one of the reasons the males need to climb out. They can get genetic mixing, because they'll just start to form one big colony from different foundresses, and that's very rare. But what it also does, they also seem to somehow be able to get all of these entry holes on a single plane. and that'll cause these fissures. Unfortunately, since 2019, branches falling have not been as common. So it could be that due to stress, heat stress or something, there was a lot of attacks, probably 2015, 2016. And then so the weakness finally showed up in 2019. Since then, I see a branch here or there, but this year there were dozens, hundreds of them in some cases all over the place. So the other symptoms, on rare occasions, they will attack initially on the bole. I went to see one a few months ago where a tree about this big had died from an infestation, had been taken down to the stump, and they could be all the way down into the roots. And the tree right next to it was also a Valio, and presumably they all came out and just, boom, attacked the bole. The symptoms of the attack in that case were very rare. The entire canopy just went boom. 1 or 2 weeks, no top-down dye, the whole thing just went brown. I was hoping to get some infested branches for some experiments from them. Waited all day, every branch was clean, the whole bowl was just full. But that's not common, but it certainly happens under the right conditions. In many cases, it might take 5 or 10 years for this to happen. This is the lower bowl. It's a little hard to see, but there's spider webs. Those can really help you out. And there's mosses that are trapping all of this very light colored boring dust. Here, if you peel the bark back, you can see all the galleries. So this is a late stage fall attack. But by the time it gets to this stage, the tree is really dead. We're going to die very soon. Here's some of this longitudinal staining. There's the attack. Fungus doesn't really go here. You can see one of those black lines where it's been walled off the longitudinal spread, but it's still spreading up and down the bum. And especially in Valio publuo, the staining from the wilt fungus is very subtle. It's just a very light yellow. Probably the darkest would look sort of like this. But here's the heartwood. On rare occasions, they go a little bit into the heartwood, and you can see there are these galleries that come right up against the heartwood. That's where the males are hanging out down in here. But what if it's cut? It's nowhere near as obvious as if it breaks off, because you don't really see the galleries that much. If it breaks off, it's just opened up. You can see like a half pipe of where the galleries are. So if you're just cutting it with a saw, it's much more subtle, especially if it is an intense infestation like that. They rarely attack branches smaller than 8 inches. I've seen it as small as 4 inches. That's very rare. And that was a very heavily infested tree. Okay, so this is one of the first that was discovered, and maybe where the first beetles, I think it is where the first beetles were collected that got identified by Sherry Smith. This is her photo. When we dissected this tree, we took it down. This is where it looked like the infestation started. That's in that branch. We got the fungus in there, and then the beetles start moving down. One of the experiments we want to do is to see what comes first, the fungus or the beetle as it moves down. We still don't know, because it would be really nice to know is like, if you saw this getting all thin, could you have gone down and thrown it here and stopped the infestation? So that's a question we want to answer. And then as it moves down, because it's a wilt fungus, the beetles can't really go back up. They can go up maybe less than a meter because it chokes off the water supply. And then they're like, oh, can't grow my fungus anymore. But, oh, look, there's water. It's like down toward roots, more and more water. So they just keep going down, and its symptoms tend to be on one side. Once they get down to where all the branches come together and they get in the bowl, then it gets girdled. It doesn't get girdled by the beetles and the cambium or the flow. It's that fungus clogs up all the sapwood, and then it'll die. There were some trees in Sacramento, the first ones discovered there, that just had one little branch that died. They went down that branch. They went fast enough that almost nothing else in the canopy died, but they got down into the bowl, and within just 3 weeks, the whole canopy turned brown. So it can be very sudden, and it can be very slow, which again makes it very difficult to notice. So we think that temperature also affects the intensity of the attack. So almost all the original infestations were out in Sonoma and Napa and Lake County. It's relatively cool there compared to downtown Sacramento. And if you're looking for the boring dust, it's fairly subtle. It really helps have the spider webs and stuff to trap it, especially if it's rained recently, it'll just wash it all away. But if you're in the middle of Sacramento, this was like 4 or 5 inches deep. And at the base of the tree, there was a pile like 6 or 7 inches deep. And I know that talking to other people who have gone out and done some surveys around Sacramento, this is not that uncommon. So I think that the higher temperatures is allowing them to really go to town on these valley oaks. And so if you're looking for the symptoms of attack, other things do produce boring dust. What you want to see is a very fine, light-colored boring dust. So I've had a bunch of people report, oh, I think I have it, but it was just all carpenter ant, which is very uneven sizes from quite large, a centimeter or more. The mob dust, if you get it under a microscope, is very fine, and it's basically exactly the same every piece because their mandibles are just nipping off the exact same piece, but it's just like a powder, few hundred microns. So I was looking to see if we could find some beetles for the managers there. We were sifting just through the boring dust. There were a lot of dead beetles in that boring dust, so it's an easy way to get a specimen. And then as I was taking that picture, like these two crawled out and another 5 or 10 were just sort of in that boring dust, kind of hanging around in there. So that in some cases, you can actually, in these later stages, get the specimens pretty easily. If you were to get specimens like this and you suspect that they might be Mediterranean oak borer, you'd want to put them in just a little bit of alcohol to kill them and then send them off to the CDFA for identification. Or you could contact me. You want. So in terms of symptoms on this tree, a week or so after I took those pictures, I came back. We had pruned off 2 branches. You can see them here. Unfortunately, it's kind of subtle. You've got some staining here from them and a little bit of staining up here and a few galleries, very difficult to see. These are the Mediterranean oak borer galleries here, and then there's some mixed in here, but the other thing that happens is these guys get in there, they start to weaken the trees, and there's 2 jewel beetles that come in, and you'll also get some others. So it's like the apple borer, and there's another one, flat-headed borers that'll join the party too. And they'll start really kicking out a lot of darker brown on a frass that also mixes in, so that can make it a little more complicated as well. This staining in the middle is this is just the typical decayed fungus getting into that hardwood. So we tried to see what would happen if we tarked it. Initially, the arborist covered 2 big piles in the same neighborhood in Santa Rosa. In a month, it had all decayed and turned into powder, basically. So this landowner went out and invested in this like $600 greenhouse tarp thingy. I said, well, I'm not sure how well it's going to work. Can we cut holes? And he's like, cut holes in my $600. So we covered it in July. I made these little traps. I tried a bunch of different ones. Ended up using these little traps that are usually used for crickets and things, but they worked pretty well under the tarp. Just taking a soda bottle, flipping it over, and I made a donut of alcohol in a little sleeve to put around it. And I was still getting them in March 24. The problem is, the thing that's going to stop them again is when the moisture level goes down below. Every time I climbed underneath this thing to check my traps, I was soaked to the bone. It never got less moist under there. You have to get that moisture out. It's not so bad with G-Soft because they're big, and if you've got the steel mesh, it's not, you can cover it, it'll dry out. With these guys, you'd have to have some pretty high-tech stainless steel, and the holes would have to be tiny. So I don't think we're going to have any luck. I think this shows a lot of promise. It's one of the other experiments we're going to try this year. I've done it with blue oaks in the valley for the Nature Conservancy. They had some legacy, huge ones that they wanted to save. Because during the droughts in 21, especially because there's a lot of heat, and it's one of the things we've learned is that drought is bad. Heat is the killer. They can stand, like in 2021, we went back and we measured it, and we're looking at them, there's no flagging, everything looks fine. But the water stress is much less. It was twice in stress. And it wasn't until September when we had a heat wave, we got a little tiny bit of flagging, and then it rained a few weeks later. So even though the water stress was way higher in 22, they just had almost no effects because we didn't have heat waves. Last year was one of the worst years ever because we started getting heat waves in May, and then we got it in little bit worse one in June, dried out the soil, and then we just had a brutal one in July. And I'm still seeing trees dying to this day because of the delayed asylum damage. But what we did is I calculated, well, it was fortuitous. We were in the middle, we were planning it. And then I was measuring all these different species of trees just to see conifers, hardwoods, just to see what the water stress was doing in 2021. And then some monsoons came through, dropped about an inch of rain, and then everything recovered. So I did a calculation, guesstimated how big a circle was around there, how much water would an inch of rain be on top of that cylinder. And so we gave about 300 gallons a night over 2 nights with just the little emitters. So we just had the brown hose with the little emitters on it. And then those trees were fine. We did that in the beginning of August, and those trees were fine all the way through to October when it started raining again that year. So that was blue oaks. So it did not take hardly any at all. They started to get stressed out, but the thing that seemed to be the most important was we were taking 4 samples around the canopy of the tree. The ones that weren't watered would be the variance on those samples was huge. The tree was allocating the water differently around the canopy. The ones that we watered, even though they started to get into that, what I call the yellow zone, right somewhere between where they're happy and where they're definitely going to start having damage. I'm not going to say the numbers. It's not going to mean anything, but there is a sort of yellow zone, and it depends. Each individual reacts differently, but they got pretty high, close to being definitely stressed, but every sample was identical around the tree. So they weren't having to allocate yet. And that seemed to be what was more critical than the actual reading itself was the variance in the reading. So we're going to try it with valley oaks. It's going to require more water since they're much less drought resistant. We'll probably go at about 1,000 gallons. So don't move firewood. Oh, and also, well, I'll get to that down there. Remove the infested trees, chip to less than 3 inches, and try not to move it, leave it on site. In Oregon, they've got landfills where they're burying them. Probably best to have about 8 inches or more of soil on top. Burning would be the best. That's not very practical. And again, like I said, watering. There have been arborists who have been injecting the trees, not with the ArborJet, but with these they started with ArborJet, but they were having the same problems with getting huge cankers all around the trees, and they were dying from that. So they switched to like the little injectors, the little like syringes, are about yay long. They go about every 3 or 4 inches. Initially, they were doing the same 2 insecticides as with iShab. Was very expensive, so they then switched to the fungicide only. A couple of weeks ago, I got some limbs off of trees that had been treated starting in about 2017. It looks like it's sort of slowing them down. The limbs still had a lot of beetles in them, but the fungus was really walled off. Thick black layer. There was a good 7 years of pretty nice looking growth on it. And the galleries didn't seem to have any fun of that, running fungus through it. So working with Mike Jones, he's got all the licensing to do the sprays. We're probably going to do a phosphide external spray because it's all we can afford to do. And we're going to work with the Napa Valley Land Trust to treat a few trees and to see if we can see any effects. But so far, they don't really seem to be very effective. We've had some other landowners that have injected trees and they died, like 2 or 3 trees. But they might be helpful, especially the fungicide, sort of helping the tree to kind of at least slow them down. And so we've got a website, just like for the GSOB and the iShab, where you can get information. You can report suspicious trees so we can come check them out. You can do the same thing, send us pictures. If you get the beetles, you can send the beetles either to me or the CDFA. Honestly, if you send them to me, it'll probably turn around a bit faster. And then I'm just going to torture everyone who helps, since this is a short list. It's actually way more than this. Between time, like this Dick Schneider with Pacific Tree Care, he was the first one to collect them, and he was just trying to get somebody to pay attention for years until people did. And then just to really torture you guys, I just wanted to put this up. You have to memorize it all for the But basically what we did is we just sequenced. Oregon sent me theirs. I sequenced them. I sequenced a few, some other people, South Conway, I think, sequenced some others, or maybe Davis, I forget. And then there was a bunch on GenBank and other places, the Bolds website. And very surprising because Oregon's like, oh, you gave us this beetle. But theirs was different. Theirs came from, like I said, it looks like probably from the Rhine Valley. They come out of the tree and crawl around when it's like 35 degrees out. You'll never see that in California. And that's a colder area than where we think they came from, probably in the Rhone Valley or something like that in France. And this shows these Portuguese ones, they're still described as the same species, but they almost certainly are not. All right. Oh, and something I forgot to mention, it was in the slide. In terms of elevation, in Europe, it's been documented. They really start to drop off at 1,500 feet. They go up to 2,000. I've found them at 1,950 on Mount St. Helena. And they're actually, at least in Northern California, there are a lot of places. So like, they call them valley oaks. I like to think of them more as alluvial oaks. So if you've got like a little hanging valley with nice silty soil without any rocks in it, you can get them up pretty high. So I've been looking, I'm hoping to collaborate with some people in Lake County to go up above that 2,000 limit to see if we're getting a difference there too. So it's possible they could up on. Let me just make sure I didn't leave you guys high and dry. Shalio quiz. Does anyone have any questions?
We're here in Southern California.
Because no one's brought any firewood down here, but they could be on their way right down. Yeah, all they eat is the fungus. So we call them ambrosia beetles, and you might think that's all one big group, but it's not. They're just bark beetles. There's like 5 or 6 branches of the bark beetle family tree. And so bark beetles have fungi. They use it to keep decay fungi out or help digest the cambium, but they're mostly able to eat available sugars in that cambium phloem zone of the tree, because obviously the phloem's got sugar. These guys, 5 or 6 times, the first one was about 70 million, 65 million years ago. The most recent one's about 20 million years ago. They got a fungus that started to allow them to go into that xylem. The fungus eats the lignin and the cellulose and the nitrocellulose, and then they eat that fungus.
So it's the side work. It's a beetle too.
Exactly. So the idea is to starve them out. And even if they're in there, since it's really the fungus that's choking the tree off, you can have the beetles in there to a certain level. As long as they're not spreading very fast or at all, maybe it won't kill the tree. But my guess is it would be a losing battle. But you might get another 20 years or so out of it. But we don't really know yet. They've done annual treatments. They've done every 2 years. It depends on the landowner and what they're willing to spend, and it's zero scientific rigor on it. So that's why we're hoping to at least get a preliminary study going this year and then get a grant to find someone at Davis or Berkeley who's willing to and has the time to do a statistically verifiable study so we can finally start to get a recommendation on it.
All right, so everyone's kind of covered the major pests and disease, all the new fancy stuff. So I'm going to cover kind of more the regular stuff that I get calls on quite a bit that you see more often in landscape and stuff that, just a little bit more common, fancy new invasives. But before we get there, we got to talk about oaks in general, which we kind of passed over. So let's start with California native oaks here. Let's name all our oak species. Okay, let's start with the white oaks. What natives do we have? Do we have valley oaks? So these are all our native white oaks that we have. Everybody can name these, right? Everybody can identify these while you guys are out there hiking. You guys can all identify these properly. Oaks hybridize really readily, so, some of these aren't necessarily always the easiest to identify. All right, what about our intermediate oaks? Less of these, so you guys should be able to get more of these. One? Somebody, what's our most common one? Nope, not agrifolia. Canyon live oak, most common one that you're going to come across. Then we got some shrub oaks, tomatella, which I'm trying to get more people to plant around here. We'll see if that ever works out. And then last one, a shrub oak that I don't think anybody's really ever heard of. All right, what about last group here? Okay, coast live oak, we can get that one, right? We haven't covered that one yet. What else do we have in this group? Black oak, all right. The 2 big ones, what else? Okay, we have Wislisnii down there at the bottom. We also have Santa Cruz Island oak. There's two different varieties of these, one that actually lives on the island. Then you have the mainland variety, and then the Wislisnii, the interior live oak. So everybody knows all these oaks, right? We all can identify them perfectly, no problems. That's like up with library. But essentially, every oak has its own set of problems, right? Every species that we've come across today, all these pests, all these diseases, they all attack certain oak species. So depending on what oak you're trying to deal with, what pest problems, it has its own set of pests and disease that you're generally going to find on those. So, hey, I'm dealing with an agrifolia. I know generally what kind of pests I can expect to find on there and what those symptoms are. So that's just agrifolia. We're going to talk about some fire stuff, just general idea. All right, irrigating our oak trees. Let's start with this. Is it a good thing or a bad thing? It depends both. Depends on when, could be both. Location, time of the year, everything, stress, all that good stuff, right? So generally, watering doesn't necessarily kill oak trees, though, right? What kills the oak trees? The pathogens, right? So overwatering weakens the trees, and then you get pathogens that come in, like we said. So the overwatering predisposes the trees to diseases. But really, in drought years, especially in our urban environments, we do need that supplemental water. We don't have deep water tables like we used to here because we're drawing all the water up for ourselves to use. So we need to provide some supplemental watering on occasion in our urban environments. But how are we actually applying that water? This is one of the most important things, that if we are going to apply water, how we're actually doing it, right? So this isn't the best graph. Obviously, our roots are extending out a lot farther on this tree past the drip line, but we don't want to water near the base of that tree, right? We want to be watering out where these roots can actually use it, where those feeder roots are, which is towards the end of the drip line and extending out of the drip line. We don't want to water essentially underneath our oak tree, because that's where you're going to get all the disease and pathogen issues. All this makes sense to everybody? This is what everybody does when they're watering their oak tree, I'm sure. But, generally speaking, that's how we want to water. Even most trees, not just oak trees in particular, but most of our trees, we don't want to be watering at the base. That's when we get issues, whether that's conifers, your pines, your, any type of tree you want to water out where the feeder roots are, because that's where the tree is actually absorbing water. Anything closer to the trunk, the water's just sitting there, tree's not absorbing it anyway, so why put that extra water there and cause disease issues? All right, pruning our oak trees. Why do we prune? Aesthetics, safety, make them look pretty. Do we need to prune? No, right? We do it because we have our own reasons to prune things, but we have this specific objective in mind. So if we are going to prune, that's what you have to think about, is what do you want your ultimate goal to be if you are going to go out and prune your trees? So if you're going out and you're just saying, I think it's the time of year to oak trees, that's what the master gardener said, or, somebody else said that this is the time of year we prune oak trees, so I'm going to go prune my oak tree. If you don't have a predetermined goal, don't touch your tree, right? Just because it might be the quote unquote time of year to prune a tree, if you don't know what you're pruning it for, if you're just going out there to prune it, don't touch it. And with oak trees, generally the decay fungi is ultimately what kills most oak trees, right? So it's not a lot of these pests and disease that we're coming with, they are killing oak trees as well, but in nature, decay fungi get in there, and that's what kills oak trees in nature. And by us going through and pruning trees, we're encouraging that decay fungi. So we do have reasons to prune, right? Hazard reduction. You want to prune out that dead wood. Obviously, we've been talking about all day pests and diseases. Pruning that out can help minimize your pest and disease issue. You also have clearance for all you street guys. Also training young trees. While trees are young, it's a lot easier to prune them and train them up properly, so that you get your clearance while they're small. And once they get bigger, you actually don't have to go back and prune them as often. As long as you started when they're small, they're more resistant to pests and diseases. So that's really when you want to focus the majority of your pruning, is on young trees, so you don't have to worry about going back and doing it later. Then obviously for aesthetic purposes, we always want trees to look pretty. We think they should have a certain form or a certain look to them, but that's, generally not the best thing. Do we all know how to prune trees? Yes, no, everybody, I would hope everybody knows how to prune a tree, right? So we have branch bark ridge and branch collar. If we're pruning this, what do we want to do with those two? We want to avoid them, right? We don't want to prune into the branch collar. We don't want to cut into the branch bark ridge. Why? Right? So this is where the majority of the tree is sealing itself. Once you cut into this, essentially you cut into the tree's potential to seal itself up properly. And now any disease that was in this branch can get into the main stem, the trunk, the bole of the tree. So as soon as you start cutting into this, the tree essentially can't seal itself off properly. And that's when you get disease progression further into the tree, ultimately causing a lot more damage. Everyone good on this? Everyone knows? All right, what about timing? I get this question a lot from a lot of people. They want to know, when do I prune certain species of trees? So when do we prune oak trees? Fall? Summer? Why fall?
Everyone don't normally see in a year when you don't want to open the, well, most oaks are linked from sunburn. You never form one in some of them exotic, obviously.
Right, we had someone say summer. Why summer?
The native oaks are dormant in the summer.
Okay, so the evergreen oaks generally, quote unquote dormant in the summer, right? So they're not actively growing as much in the summertime.
Less chance of
Less chance of possible spread during the summer because it's warmer. According to California Native Plant Society, this is their recommendation. You prune deciduous oaks in the wintertime and evergreens in the summertime. So what do you guys think about this? Yes, no? We don't have fall on here. What do we think?
We're putting sunburn.
What about, it's true, oak trees, are Quercus agrifolia is susceptible to sunburn.
It's strawberry, not the first in the land.
I don't necessarily agree with this myself. Well, what are the advantages for pruning deciduous trees in the wintertime?
You
can see a lot better what you're actually pruning out, right? Gives you a better idea of what's going on. You also have a lot less mess to clean up. So you don't have all those leaves and everything that you have to get rid of. So some advantages there, but it's wet, generally speaking, right? Wintertime is generally when we're starting to get rains. So that could cause some disease issues. Does timing really matter at all? Is there ever a good time to prune oak trees or prune trees? Probably not. Generally speaking, I think there's not necessarily one time of the year that's better than others. Couple things to consider. This is canker pathogen growth on pines, not necessarily oaks, but you can see some bog cankers here. And generally these bog cankers are, much more active during warmer weather. So if you're out there pruning in the summertime, you may introduce a bog canker that is actively growing and can potentially establish easier because it loves those warmer temperatures. For pruning in the fall, that's when our decay fungi are typically throwing out fruiting bodies. So you're going to have a lot more decay fungi spores in the air if you're pruning in the fall. So, there's all these things to consider when you are out there pruning. And, I really just think that there's not a great time to ever prune oak. So you just want to manage it as best you can. And, if pruning isn't necessary, don't do it. All right. So I was asked to present some stuff on oaks and fire. I've been out dealing with the fires quite a bit in LA area the last couple months. So I thought I'd throw this in here. Generally, when we think of oak trees and fire, how do we think of our native oaks doing?
Exactly.
Pretty good, right? We generally think, fire's native to California, so our oak trees are generally fairly adapted to fire, moderately anyway. And what that has to do with, generally speaking, when we talk about oak resistance to fire is how thick the bark is. And that really determines how well oaks can handle fires. What are our kind of native fires in California? Are they grass fires, chaparral fires? Are they quick moving, slow moving? Generally, what do we have in our oak lands?
Fast moving fire.
So they're generally fast. They're burning that understory fairly quickly. So it moves through fairly quickly. And, generally there's not hot fire per se burning our oak trees. So they can withstand that moderate amount of fire because the fire comes in fast. It moves through, it burns that understory, all that grass, and just keeps moving on. And here you can see different bark layers, how thick the bark is on all our different species. Just a few species here in California. So which species here is going to be most adapted to withstanding fire? Agrifolia, right? You can see the agrifolia has a lot thicker bark than any of the other species that you're typically going to find in California. So the agrifolia is generally considered a lot more fire resistant than the other species we have in California. Some of these can do okay, and some of them, even though, like Chrysolepis here, the bark itself is actually somewhat considered flammable, and the bark on this fire will kind of travel up the stem on the Chrysolepis. So, even though we think our native oaks are fairly fire resistant, they were adapted to a certain type of fire, and our new fires don't deal the same way that the fires did when they evolved. So here's a picture of the Eden fire, the coast live oak and the Eden fire. You can see the oak tree is sprouting back out green. There's a lot of new green in the canopy, but when you actually go and look at the cambium health down below, where that structure burned, where that house burned, fence right here, that was about a foot away from the trunk of the tree that burned. On the far right there, you can see the cambium on that oak tree is completely dead. And here you can see where bark burned. Underneath that, the cambium's dead, and you have live cambium next to it. But on this tree here,
I got some potatoes.
oh, I don't want sweet potato. Sweet potato. No.
Yeah.
But on this particular tree, there was over 50% of the cambium dead that's on these.
No,
the other one I like. So survival of this tree in an urban environment is a lot different than in native habitats. 50% cambium. Obviously, you can see trees sprouting out. Essentially, the tree, will survive. But you want a tree left, 10 feet from your house, that 50% of the cambium is dead on. Is ultimately some of the decisions that we have to deal with in our urban environments that you don't necessarily have to in wildlands. So, oak doing okay. They couldn't withstand that high heat, even though, it's our most fire resistant species, but just the high heat of those buildings burning still killed the cambium on here. Here's another one. This is in the Palisades fire. Very similar reaction to it. There was a fence that was on the outside that burned. And here you can actually see the bark starting to crack. So the bark itself is now dried out. Bark is cracking. No cambium left alive underneath a lot of it. And then you have oozing. You have this bleeding going on that oak tree as a response to the fire as well.
That thing's like grass.
Here, there wasn't frass on this one. I have seen some frass starting to form out there, but not too much yet. One thing that I did want to point out on this last one, on that picture on the right, you can actually see some fungus starting on that bark that's burned there. You can see that greenish gray color, and seeing a lot of that now in the fire on trees that have burned in the fire. So we'll see what happens with that, but starting to get some fungus that's moving in on all these fire damaged trees already. You can see them fruiting and thriving on that fire damage. All right, getting into our pests, our less common stuff here. Any questions on the fire stuff before I continue? Before we move on, Mike? I don't. Oh, he asked if fires like this have killed any beetles and stuff. I am just starting to find very slight evidence of new beetle activity post-fire, but looking at ISHB and some of those other beetles, it's hard to say because I didn't have any plots or anything out here, pre-fire to know how they're doing after the fire.
So Kim Carella's doing some very on plots on GSOC down in it. I don't know what her results are, but I'm also doing them up north. And we're looking at everything attacking them after the glass fire. And for the first year or two, there were some bugs, but after that, they kind of went away. And the trees kind of got their resistance back. So in general, if the tree burns enough to kill the bugs, the tree's dead.
Yeah, I mean, that's generally what I would think too. If you know, for a lot of these beetle species, they're fairly far in, you know, they're in the cambium layer. So if it's getting hot enough to kill the beetle in that cambium layer, it's going to kill the cambium of that tree as well. But, you know, maybe some of those other bark beetles, some of the minor bark beetles, it may be effective on those, but
yeah, I mean, in the literature, it's usually an elevated amount of insect activity post-fire.
All right, what pests do we have here? Mistletoe, everyone knows this one, right? So this is broadleaf mistletoe. There's quite a few different species that we have in California. It's a parasitic plant, relies on water and nutrients from its host. It's dioecious, which means what? Male and female, right? Male and female. So you have a male plant and a female plant. It reproduces by seed. Here's general mistletoe life cycle. So you have these birds that love to feed on the seeds, on the berries. Then they goes through the digestive system. There's a sticky coating on the outside of the seed that sticks to the branch surface. That seed germinates. You get the hypocotyl that goes down, attaches to that host branch, and infects the host branch. And then you get this Ostoria that infects that branch and spreads out. And over time, you can actually get new plants that germinate from within that Ostoria that's infecting that plant tissue. Damage, generally we don't think of mistletoe causing major damage on our trees. You know, they are feeding off the tree, right? So they're stealing those nutrients and that water from the tree. So they are weakening those branches. And in severe cases, they can kill a branch, but generally they're just reducing the vigor and stunting the growth of those branches that they're on. You can also get witch's broom at the infection site, but generally we don't think of it as too big of a deal. Management, generally your best option for mistletoe management is mechanical control. So you want to prune out, you know, if you are worried about it, you want to prune out your branches at least a foot away from that attachment point. If, you know, you can't do that, or you have other options, you know, it's a branch that you don't want to cut, you can prune it out where it's where that attachment point is, and then wrap it with black plastic. And, you know, even if you cut off the plant, that it's just going to resprout from that Ostoria. But if you cover it in black plastic, eventually over 2, 3, 4 years, that will kill the mistletoe, but it's going to take years. So it's not a quick process, not something that's easy to do. There are chemical control options. Fluorel does work great, causing the plant to fall off from the tree, but mistletoe resprouts again from that site. So it's something that you're going to have to do a few times to actually get control. It's not just one application. It falls off. You're going to have to do it every time it resprouts. Mistletoe does take a couple years before it starts producing berries and seeds. So you can, if you want to go out, you know, treat it when it resprouts next year, treat it again, and eventually it will, you know, you will kill it, but it's going to take a long time. There's reports of glyphosate being used on mistletoe and not working effectively. Glyphosate is not registered for control of mistletoe, so I'm not advocating its use. I haven't tested it or anything, but there are reports of it working. In host resistance, conifers are much more resistant to broadleaf mistletoe, although then they get dwarf mistletoe. There's also some work on looking at transgenic species for mistletoe resistance, but some species just naturally have more resistance to mistletoe, and they're trying to play around with, you know, how mistletoe actually infects branches and things like that, and seeing if you can get, you know, turn on certain genes to actually cause resistance to mistletoe. But, you know, nothing set in stone right now, but there are researchers playing around with that kind of resistance. All right, what do we got here? Got some small holes here. What if I showed this one? Gall wasps. Oaks get a ton of galls. There's lots of things that cause galls on oaks. You know, they're this kind of foundational species in the environment. So there's a ton of things that will cause galls on oak trees. Generally, we're mostly thinking about wasps causing the galls on oak trees, but you can get it from mites. You can get it. There's a gall fly as well There's a lot of things that cause galls on oaks. What do you guys think? Are they a big issue? Not an issue? They might be. We're going to talk about something later on something that I was dealing with this week with a couple people in the crowd. But what do you guys think? Issue, not an issue? You guys treat for it? Don't worry about it. You know, there are some pretty cool galls out there. I mean, you get like the apple gall and the fuzzy galls, so they look cool. But generally, we're not treating for them, right? All the research that's been out there, you know, chemical control is not very effective on them. So, you know, you can try, but, you know, they're kind of feeding, they're causing this corky layer a lot of times. They're causing these trees to react differently than they normally would. So it's hard to get the chemical in there to actually effectively control any of these gall makers. What do we got here? A shiny beetle. Yes, a shiny beetle. Anybody know? Nope. This is oak twig girdler. So it's another agrylus species. This is oak tree girdler, twig girdler. Feeds on small branches, girdles those small branches. Takes about 2 years for these beetles to develop. So they're in there feeding for a couple years before they become adults. Adults generally emerging during the summertime. And they're there. You see it all over Southern California. You'll see dead tips on branches and stuff. Now you can go to Disneyland, see dead tips on branches from this. But generally, not too significant. You know, you're not too worried about a few small twigs dying here and there from this guy. What about this one? Bark moth, clearwing moth, sycamore borer. I think this is a really pretty insect. Looks really cool when you actually are able to find one. But sycamore borer, most common name for this one, feeds on the inner bark, causing this formation of cork cambium. So you get this really corky bark, this really gnarled bark on the trees. But generally, it's not causing a whole lot of damage to them. Management. How many of you guys treat for sycamore borer? Anybody? I know a few people that go out and they used to treat preventatively for sycamore borer and had pretty minimal effect, but there are beneficial nematodes out there that are effective, but it's really a pain to apply them properly to cause effective control. So if you are going to do beneficial nematodes for sycamore borer, essentially what you have to do is put it in like a little bulb or application, and you actually have to squirt the nematodes into that gallery of the sycamore borer to get good control on them. So it's labor intensive, although, you know, they are effective. It's just labor intensive to do it properly to get good control. There is pheromone traps out there for other species of clearwing moths. We don't have any for our sycamore borer. If any of you want to go out and collect pupa for me, USDA said they would actually create a pheromone for sycamore borer. But I have to send them pupae. So if anybody wants, you know, a little project to do, go out and collect a whole bunch of pupae and we'll create a pheromone for this. You want live ones? Live, yes. So essentially we need virgin females to create the pheromone. What do we got here? Kind of looks like a sphinx moth. Anybody know what pest this is on oaks? Generally, I see this as more of an issue in Northern California than here in Southern California, although it is here in Southern California. But you get more reports up in Northern California. It is not a leafy feeder. Interesting. Carpenter worm. How many of you deal with carpenter worm? Generally not too often down here in Southern California, but it is here. But I hear a lot more reports of this from Northern California, from colleagues up there. But it can be pretty devastating. It does feed all the way through into the hardwood on oak, so you can get branches that break and things like that. But this is carpenter worm, causes a fair amount of damage. Management wise, they like stress trees, so just keeping your trees healthy. Once again, we have beneficial nematodes that work for them, but same thing, you got to get those nematodes into those galleries before they're effective. So not the easiest. And then chemical control, you know, it's really about timing and trying to get those pupa. So those pupa will come out to the bark and kind of stick halfway out of the bark. And so if you can time it properly, you can treat those pupa on the bark, but not that easy to do. But because they do bore into the hardwood and stuff, if you do have, you know, fairly heavy infestations, you want to remove those branches out so you're not getting branches that'll break in the wind or anything like that. Paul?
Do they have a preference for oaks or what species of oaks or all across all of them?
It's quite a few oaks. I mainly see it on agrifolia, or I mostly get reports on agrifolia, but it'll attack most of the oaks we have in California. All right, what about this guy? What do we got here? 2 years ago, I was getting calls about this guy all the time. When I was first starting with A&R, I was getting a ton of calls for this because, it was still kind of drought, and these were really going after the agrifolia down here. They'll attack a few different posts, like the Pacific fly-headed borer. So these really love stressed oak trees. And so when we had the drought and the heat wave a few years ago, these were really thriving on oaks. And I was getting calls from arborists, that had been in the industry for 20 years. And they're like, I've never seen this pest before. Is it something new? And now I'm like, no, our native borer here, they just really love those stressed oak trees. All right, so we're getting ready to wrap up here. I'm going to finish this up with a little kind of Q&A on a pest that I went out on oak trees that I was looking at this week. So this is the LA Country Club. And what do you guys think's going on with this oak tree? It's hard to see in this picture on here, but you have dieback all throughout the tree. What do you guys think's going on? Could be so many things, right? Everything we talked about today causes this, right?
Is it on a slope?
It is on a slope, but here's another picture when you look a little closer. What do you guys think's going on here? This is at the LA Country Club. You got this weird girdling going on the branches.
Is that natural or
Nope, this is what it looked like when I came up to this tree. And this is weird. Yeah, bark's gone. There's no bark at all left in that. Could be mechanical, could be some kind of rodent. Here's another branch on that same tree. You can see a bunch of gall wasp on there. And then here's how that starts. And here you can see teeth markings where, I don't know if it's a squirrel or a rat, looks like it's going after those gall wasp larvae. You can see those teeth marks kind of pulling up all that bark, trying to get out what's underneath. And here's what was underneath that. You can see all the little larval pits right under there that those larvae were already gone. So it had pulled off that bark and was feeding on those larvae underneath it.
So how big are those pupal chambers?
They're
Like a quarter?
they're small. I mean, you're talking what, a little bit bigger than the tip of a ballpoint pen, maybe.
Is it not? So that's just a tiny twig.
These are fairly sizable branches. I mean, they're decent size hits, but they're not huge or anything. And then here's another oak on the property that has just a lot more girdling on it. And this one, there was no obvious oak gall wasp or anything on the trunk. They just seem to, whatever, at the golf course, whatever pest it is that's feeding on it really likes feeding on oak trees now.
Ground squirrel.
Some kind of rodent, but it also looks like there's some kind of secondary pathogen moving in and all this damaged area as well. But yeah, some rodent damage as well for oak trees. I know I'm missing a lot of people on here. Work with a lot of different people, just like everybody else. There's a workshop tomorrow on fire stuff. If anybody wants to log in on Zoom or join us in person at the LA Arboretum, learn about how trees are doing in the fires and all that. Yeah. Oh, we're back.
There's this few questions in the chat. I think this one was about mistletoe. It is, what is an acceptable tolerance for infestation before tree removal? We have 3 trees with more than 15 clumps per tree. It's concerned about spreading other trees.
They are going to spread, so that's, question is how much mistletoe is an acceptable level, and it's really up to you as the consumer what you're comfortable with. I mean, if you have 15 different spots in a tree, that's going to produce a lot of seed, a lot of berries. That's going to start spreading to any other trees you have nearby. So, at that point, do you care if the mistletoe's there or not? Really, I don't see a whole lot of branch death per se on a lot of our oaks around here because of mistletoe. So it's really up to you whether you can tolerate that mistletoe that high of a level, but it's going to be spreading around with that much mistletoe in your tree.
Yeah, one was your cats.
Okay. Yeah, probably be offering more of those. I am going to be offering more post-fire tree assessment trainings and workshops. We're just trying to get some training out there sooner rather than later. So we are going to develop better course material and stuff for post-fire material, but this is just the starting point to get the word out there and for arborists that are actually going out there in fires and aren't necessarily the best trained on what to look for and things like that. I think that's awesome. Like we will.